And it makes me really happy. Like, it's really nice to see everyone together. I'm super excited to introduce our speaker, Dr. Amanda Whitmire, and I'll tell you about her credentials and all that in just a minute. But Amanda was invited by me, Rebecca Kim, and Natalie Lowe. Kind of, we all wanted to invite Amanda separately. Natalie actually knew Amanda, like they worked together. But Rebecca and I more were like Twitter friends of Amanda, and maybe stalkers a little bit, following all of her amazing work. And so we're just really thrilled that she could be here today to talk about the work that she does and talk with us about the way that the work we do here at the Academy intersects with the kind of things that she's trying to do at the Hopkins Marine Lab. So let me tell you a little bit about Amanda. She's the head librarian and bibliographer of the Harold A. Miller Library at the Hopkins Marine Lab, which is part of Stanford University. She has a BS in aquatic biology from UC Santa Barbara and a PhD in oceanography from Oregon State University. She is really, as you'll hear, an oceanographer turned librarian, and she has a passion for rescuing old data. So, right, you can see, like, so do all of us, right? So, and she believes that observations from the past are critical to being able to put current changes into context. And so welcome, Amanda, and we're looking forward to hearing all you have to tell us.
Thank you all so much for coming. I'm nervous all of a sudden. It was a full room. Oh my gosh. I am so thrilled to be here. I'm having so much fun today visiting the collections and getting a chance to talk with all of you. I have to start by saying thank you to Rebecca and Natalie for inviting me, as a librarian who works at the intersection of marine science and biodiversity and historical ecology. This is being able to come to the Cal Academy is just, it's a real thrill. So thank you so much for having me. So I did not set out to be a librarian. I wanted to be a teacher. I wanted to be a scientist, but the traditional academic route didn't really turn out to be a good fit for me. And it was just sort of by chance that I ended up working in a library and then becoming a librarian. And I'm offering a little bit of context because I think it's sort of knowing my path might resonate somewhat with the students or maybe some of you. So my title is head librarian of Miller Library, which is situated literally at the end of the rainbow at Hopkins Marine Station. And my main job, the overarching purpose for my job is to support the research needs of the Marine Station. So, and not only the Hopkins community, but the broader Stanford community within the marine sciences. So the Marine Station itself is home to just 11 faculty, about 20 graduate students, 13 postdocs, and various staff. So it's pretty small. But like I said, I also support marine science happening on main campus. I manage a branch library, which means that I manage a facility on a day-to-day basis. I have to make sure it's safe and comfortable. There isn't rain coming through the ceiling. We have everything from 3D printers and Arduino kits. Thank you to Natalie's inspiration for our makerspace, which also has crochet hooks and yarn and sewing kits. I provide direct support to researchers on everything from lit reviews to data management. Librarianship is a very broad role these days. And so from day to day, my job is extremely variable. And one of the luxuries of working at a place like Stanford is that, within the purview of my role in supporting the research mission of the university, I get to sort of pick and focus on what I'm most passionate about. And in this case, it turns out to be mostly data and collections. So that leads to the focus of my talk today, which is sharing some of the work that I've been doing at the Marine Station to curate and make actionable some of the historical research outputs of the Marine Station. So the goal for the talk today is really just to share a little bit about what I've been up to, hopefully generate some conversation around historical biodiversity data and collections, and work toward building a community of practice around these things. If it turns out that any of us have interests in similar things, I'd love to just start some conversations and see where it takes us. So given my personal mandate and professional mandate to support the research needs of the station and beyond, a major question is, well, what do my researchers need? What do I need to provide them to support the work that they do? And I think it's pretty well understood probably in this group that there is a lack of ecologically relevant observational data, especially in marine environments, because it's a hard place to get to do research. So we also understand that this is like the perfect crowd. In order to be able to assess long-term changes, changes in biodiversity as a result of climate change, things like that, you really do need data over those long time periods and broad spatial scales, depending on the kinds of questions that you're asking. And Hopkins Marine Station has been located in the same place for over 100 years. So we have stuff in our collection, I think that's really well suited to filling some of the gaps in observational data that we have. And not just through digitization, but also transformation of these collections. So that's what we're really trying to do. And I'm just gonna sort of ease into exploring this idea around a lack of adequate data by taking a quick look at one of the world's largest biodiversity catalogs, GBIF, Global Biodiversity Information Facility. Many of you are probably familiar with this, but just in case, this is an international network and research infrastructure. It's co-founded and co-funded by several world governments, and it really is aimed at providing anyone, anywhere, data about all types of life on Earth. They have over 2 billion occurrence records now in the GBIF catalog. So one of the most valuable things about the catalog, from my perspective, is this collection of species occurrences, right? I saw this thing at this place at this time. And if we look at, an occurrence page for a very common marine species, this is an intertidal anemone. You'll see that there are 8,617 occurrence records in GBIF, which really sounds like a lot. Like that looks like pretty good spatial coverage. That's a lot of occurrence records. But if you dive one step deeper into the data in GBIF, what you find is that the data are heavily skewed toward the last decade or so, right? And that doesn't mean there are no observations going back in time. There's a blip there in the 70s, and they do go back into the early 1900s. But it would be really great if we could fill out the left side of that plot a little bit further. I think in this crowd, can anyone guess what might be causing this exponential increase in observations here? iNaturalist, yes, exactly. So of the 8,617 occurrence records, 8,233 are from iNaturalist, right? It is incredible. And I don't have time to go into iNaturalist for my talk today, but I love iNaturalist. It's amazing. But don't you wish we had it going back, right? So before I get into a few examples of the kinds of things I'm working on, I just want to explore something a little bit with you about how I became interested in using computational tools. And it really started to happen when I realized that in order to provide my community of faculty and staff and students with the collection materials that they would use, that I'd have to transform as much of it as I could into formats that they would use. Scanning is a first critical step, but it's very unlikely that anyone I know at Hopkins is going to use and take the time to convert 23 years of scanned oceanographic data into data sets, tabular data that they can actually use. Like maybe they would hoodwink a student into doing it for them if they were desperate. But it's not likely that they would use it. We put so much work into scanning and curating and creating metadata. And I just thought, how much more work would it be to transform these scans into actionable data, and what tools are out there to help me do that? And this is just something that I want to emphasize because it's a subtle point, but I think it's important, especially in this group, that what I'm embracing at the Marine Station and Miller Library is increasingly known within sort of the librarian circuit as collections as data. So one of the core principles of treating your collections as data is that collections as data designed for everyone serve no one. Stewards should be intentional about who their collections are designed for, work to lower barriers to use for people in those communities, and continue to assess these needs over time. And this shift in thinking, this evolution in thinking about how librarians approach our work is something that I personally think is critical to maintaining our relevance and maintaining our ability to support researchers moving forward. So if there's one thing I'd hope that you take away from my talk today, it's that, it's not actually the AI or the examples that I'm going to show. It's that the sense that we as cultural heritage professionals should really be interrogating how we're processing our collections and how we're offering them out to the research community. Sometimes scanning is enough. Sometimes that's the best we can do, but sometimes it's not. And when it's not, I hope we can all be curious about what tools we can use or what tools we can develop to get the transformations that we need for our research community. So let's get into some examples. This is the fun part. So this first example that I'm going to start with, I think should be intuitive to hear because to everyone here, because I suspect that we've all seen, if not created, a thesis or a dissertation. This story starts with a dissertation and it continues to this day. And some of you may be familiar with this work. In 1931, a graduate student at Hopkins Marine Station, Willis Hewitt, started a time series transect in the intertidal at the Marine Station. And the transect was 105 yards long. And he sampled that transect and counted the 90 most common species along that transect from 1931 through 1933. And his dissertation is in our stacks at the library. Then in the early 1990s, 1993, 2 undergraduate students came to the Marine Station to take a spring class, and they decided for their research project, they would find Hewitt's transect, which had been lost to time, but the brass bolts were still out there in the intertidal. They would find his transect and they would resample it, as many as they could within the limits of a 10-week quarter, and see what they would found. And it turns out they discovered that species abundances had shifted to warmer water species, that is to more southern species relative to Hewitt's work in the early 1930s. And Hopkins also has a time series of temperature that goes back to 1919. So they were able to correlate the shift in species that they saw with a temperature shift that was also observed at the Marine Station. But a small student paper doesn't give you much of an opportunity, so they actually came back the following year, which for our undergrads is not uncommon. Once you get them out to the coast, they always want to come back. They gathered more data, they wrote it up and ended up publishing a paper in Science about the effect of climate change on the intertidal at Hopkins, which is pretty phenomenal. So Rafe actually continued sampling the transect for his dissertation. He went on to study it for his PhD work. And then after his career took him to Duke and to Arizona, he kept coming back to Hopkins and sampling the transect. And it's still being sampled to this day by researchers at the Marine Station. This is actually Natalie here sampling just a few years ago. So the dissertation is in our collection. Their student paper is in our collection. And I started to wonder, what would it look like to treat the Hewitt thesis as data, since it's foundational to the work that we're still doing at the Marine Station. So there were actually a couple of things already in our Stanford Digital Repository. There's some digital photographs that they take of the quadrats every year, and a shapefile, a GIS shapefile of where the transect is located. But I took my work to the actual dissertation itself. And the first step, of course, is to scan their dissertation, which we did. That's the fastest part. And we also had a scan of their student paper, which is part of a different project that I'll get to a little bit later. So remember that one of the central principles of taking the collections as data approach is lowering barriers to use. And our researchers work with spreadsheets. They work with tabular data. So we looked at how we could extract the data from the thesis and offer it as a standalone data set. And before I move on, I want to acknowledge my partner in this work, Melissa Tabra. She works part-time in the library on our staff. Our digital collections projects are really only possible because of the work that she does. She is extremely diligent and detail-oriented, from scanning all the way through transcription and metadata and uploading to the repository. I rely on her to get this work done. I just appreciate her so much. So for the Hewitt dissertation, we ended up extracting 19 data tables, a list of species at the end that was provided by Hewitt of everything that he saw, a section of the thesis that was concerned with the breeding habits of some of the intertidal invertebrates, which I have not seen of continuing interest, but what that section does have is a list of species with dates and where they were seen in the intertidal, and that's the good stuff, right? Like that's what we're looking for. And so yeah, I didn't really emphasize this when I started talking about the transect, but like you get the feeling that the value of this is that this dissertation is providing important baseline data for the time series that's still going on. So then once you get the data out, like that's awesome, that's a great start. But I thought, what else could I be doing with this that would be useful to my researchers? So I just started poking around. And I know that researchers who are interested in historical ecology and species shifts and range shifts and so on, you know, they're interested in taxonomic information. They want to know that what we have in the dissertation connects to what they're seeing now. So I compiled a list of all of the species that Hewitt saw. I deduplicated everything. And then I discovered this tool, which some of you may have heard of and used, Global Names Finder through the Global Names Architecture, which is a set of tools for doing taxonomic work with text. And you can give Global Names Finder a list of names, and it will even be able to find misspelled names within reason. So if you have OCR errors or transcription errors, Global Names Finder can deal with that. And it will match the names in your list with authoritative databases, taxonomic databases. And this is the output that you get on the web interface, but there's an API, so you can use R, Python, or whatever you're comfortable with using. So here's a view of that same data as a teeny tiny text file. Sorry about that. So the verbatim name that we provided is on the left. So that's straight from Hewitt. There's a column with the quality of the match. These all happen to be exact, but it could be fuzzy. Could be no match. Here is the vernacular name, canonical name. If it was matched up with a taxonomic database, it provides you with the ID, and then the name of the database and so on. Very handy. And I thought, what else can I do with this data? So I pulled it into OpenRefine, and I don't have time to go into OpenRefine, but if you're not using this tool, it is so fun and it is so powerful. So I pulled the spreadsheet from Global Names Finder into OpenRefine. And what this allows you to do is take advantage of some of their reconciliation tools, which is a way to connect your spreadsheet again with external authorities. So in this case, I was able to connect with GBIF and get the GBIF ID for the taxonomic names. I was able to get the Wikidata ID for the names, and also the WORMS, which is the World Register of Marine Species. It's one of the marine taxonomic authorities. So really starting to connect Hewitt's work and what he saw with the online sort of knowledge graph for biodiversity. And then of course you can export that from OpenRefine and attach it to the dissertation or to the data set we pulled from the dissertation as a standalone reference for researchers. So just to summarize this example, you know, instead of scanning the dissertation and posting it to the repository and calling that the end of the day, the scanning was really just the first step toward treating the dissertation in a collections as data kind of way. So we extracted data tables from the dissertation, we enhanced some of the data from those tables with authoritative taxonomic information, and then put that all together as a standalone data set. So that was one of our first sort of forays into the world of collections as data. The next example I'll talk about is from the Stanford Oceanographic Expeditions. This was well, we all call it the Tavega cruises because that's the name of the ship that they went out on for most of the project. This was a science education effort that ran for about a decade, where they took graduate students out on ships which were used as floating classrooms. So they would go out, they'd sail the Pacific and Indian Oceans, they'd do research, they'd write up reports. You know, it really was quite an experience for the students. And we're only partway through the scanning and accessioning process for this collection because it's a really big, well, in my world, this is a big collection. Now that I've been here, I can't say that. But I couldn't resist sort of playing around with some of the files because it's just, as an oceanographer, this is just like gold. So here's just some examples from the collection. There are, you know, it's highly variable from cruise to cruise as the chief scientist changes, the way they do things changes, of course. But there's reports and scientific reports about what they did, narratives from the chief scientists that describe day-to-day life on the ship. When they went and visited stations, they do station logs, biological logs, net tows, there are species lists and so on. It's a really diverse collection. But again, I'm sort of wondering how can I use computational methods and online tools, open source tools to get at this collection a little bit. Sometimes you really have to sift through text to find species occurrences, but sometimes you get lucky and they put it all together in a species log for you. And so that's what this is. In this case, this is a list of all of the mollusks that they collected during the third cruise of the Stanford Oceanographic expeditions, where they went to the Indian Ocean. And you can see that there's a list of taxonomic names. After each taxonomic name, there's a list of stations that they visited or where they saw that particular species. And then at the very end of the document, there's a list of stations that provides the latitude, the longitude, and the date that they were there. And sometimes there's a description about that. So I had these physical materials in the library, scanned it, of course, and then the next step, as with the dissertation, is getting the taxonomic names out, right? And since this is typed, it's pretty straightforward to use FineReader or Tesseract or some other OCR tool. So with the text in hand, again, we return to Global Names Finder because it's so handy and effective and fast. I didn't mention that. This tool is fast, amazingly fast. And for this one species list, for this cruise, it found 565 unique taxonomic names, 645 in total because some of them are mentioned more than once. And there we go again into Open Refine, doing all the same steps, linking up with GBIF and Wikidata and all the authority databases. In this case, what I thought I might do is use Open Refine to pull the Wikidata IDs. And the cool thing about Wikidata and Wikipedia is that they often have more descriptive information about species. They'll have photographs, lots of rich information that doesn't necessarily fit into a database. So what I did in this case was I pulled the Wikidata Sparkle endpoint of the photographs. If that particular species had a photograph, I pulled the URL for that photograph. And why would I do that? Well, I pulled the URLs into Palladio, which is a tool from the Humanities and Design Research Lab at Stanford, and I used their gallery function to get a quick look at the diversity of mollusks represented in the collection. I am not a taxonomist. I am not familiar with the hundreds of species of mollusks that they found on this cruise, or with like any species mostly that are out there. So for me, this is an error checking step, right? Like not all of the species have pictures, and that's fine. But for the ones that are there, if I saw like a squirrel go by or a tree, like something that was obviously not a mollusk, it would clue me in like, oh, maybe I need to go back and check on this record and see, was there a transcription error? Was there an OCR error, and so on. So there are cases where I'm using computational tools, APIs. I don't know if you call that a computational tool, but using the tools that are available to just help me a better curator of the collection. And it's pretty fun. So going back to this one, oh, that's funny. There it is. Do you love how I left that in there? That was a note to myself. That's so funny. So I'm going to talk about a project I've been working on for like 4 years. It's a really fun project, but it's a really hard project. And I'm still having a lot of fun with it, but we're still trying to make progress. So this is a project that's really aimed at trying to fill that gap, both back in time and across like about this spatial scale is what we're looking at. So Rafe and Sarah's work with their student paper from that spring of 1993 really got me thinking about this collection that I have in my library, the student papers. Their paper is one small slice of a very rich history at the Marine Station of this spring class, where undergrads come out and they do very focused research under the tutelage of faculty. They're very closely monitored. And I have this collection going back from 1963 to 2011. They stopped teaching the class. Like, why would you do that? But I have all these papers in my library. I thought, gosh, how could I be leveraging these? And then it turns out that an undergraduate student paper collection is not an uncommon thing for a Marine Station to have. So my colleagues up at Friday Harbor and up at Bamfield in Canada have thousands of papers that go back to the 1920s. My colleague in Bodega Their collection is still growing. It goes all the way back to 1928. She's got thousands of papers, right? So the thinking was, if we could imagine being able to get all of these digitized and coming up with a way to sort through all of this text and find species occurrences. Like, how incredible would that be? A corpus that follows decades and decades of time over the whole West Coast of the United States. It's extremely rich. What do I want to say about that? Oh, it took me a long time. I'm slow on the uptake sometimes. It took me a long time to figure out, like, I was an undergrad once, like, I wasn't always so careful with, how valuable could this collection really be? And I think the thing that's worth emphasizing is that, the hypothesis in their paper could be wrong. The question that they were asking might not still be relevant. But the fact that they were out there on June 3rd of 1945 and they saw a Pisaster or whatever, like, that's still true, right? Like, that's a fact we can use. So that's the kind of information that I want to pull from these papers. It's not like what was the research that they were doing. It's what did they see out there? What was out there at the time? So I really think a collection like this, each of these in their own right is important, but if we can leverage across these collections, that would be extremely powerful. So that's why it's 4 years in and I'm still like, how are we going to do this? But let's talk about the approach a little bit. So, things obviously always start with scanning, and I hate to skip over that because that's a whole process, as you know. There's a lot of work that goes into that. But given the time today, I really want to spend more time sort of talking about what happens like after you scan it. And what do we do next? And this is the problem that we have. I literally randomly pulled out one paper from the stack. I clicked on one file to look at. It turned out to be this really cool paper about chitons. So we'll use that as our example. So keeping in mind our example from GBIF, the one thing we're really interested in is species occurrences. So we need a date, a location, and a taxonomic name. Like, those are the three things we're really looking for. And sometimes in papers, and I've been noticing the methods section, you will see them together, right? Like, we were out there, spring of 74, Muscle Point, Pacific Grove, Malpalia muscosa, and there's some extra information about habitat, tides, maybe you could scrape that. So sometimes the information is all right there for you, which is great. Sometimes you only get part of what you need. Like, this is a really cool list of all the things that they saw out there, except one of the sentences says, we didn't see these things. Critical difference. But there's actually some more specific date information here on this page. So that's like, okay, some of the stuff is in there that we need. But the question remains, like, as a human, you can scan over that page and you know what those sentences mean really quickly. Like, you can tell, yes, that's a species occurrence. No, that's not. How do you get a computer to make that decision for you? That is the challenge that we're facing. So maybe like we can use entity extraction. So that's one thing. Like, just give it a list of scientific names and tell me if any of these scientific names are in this text. That's kind of what Global Names Finder does. So I just put this one paper through Global Names Finder again, 35 taxonomic names in this one student paper. It's pretty surprising. I thought, all right, I'm going to put all the student papers through. So I did. This is what the JSON output looks like for one particular record from Global Names Finder. But I ran 445 student papers through Global Names Finder. It found 10,622 unique taxonomic names. Pretty cool. And if we look at a breakdown of the matches, 70% of them were exact matches for the taxonomic name, which I actually was kind of surprised by. 185 of them were fuzzy matches, which means there was maybe a misspelling, so not exact, but close enough that they could determine what we were looking for. And then 25% of the matches were no match, which seems like that's kind of weird. What does a no match mean? It turns out it's actually the abbreviated binomial name is not considered a match, but it's like we can there are tools for figuring out, what they meant by that, which is great. So, I mean, that's like the large majority of names, it's actually matching pretty well. And then, of course, if it does get a match, it'll link out to, you can select in Global Names Finder, like, I want you to use Catalog of Life and Worms, but not ITIS or what, you can choose. I just have it search everything because I don't know what's what. I'm just a librarian. And then, of course, you can pop out to Catalog of Life and get all the, vernacular names and synonyms and Bayesianonyms and all that really rich data. And then I didn't actually get to show a Wikidata and Wikipedia page. So I just love Wikipedia and Wikidata so much. So I had to throw a slide in there. And of course, there are APIs for getting all that data down. And then of course, GBIF. I really love that we can tie into GBIF with APIs, because I think one thing we've considered in helping us sort of make that decision about whether or not this is a species occurrence in the papers is, as part of our workflow, you could compare the location you get in the student paper with the, sort of set of locations from GBIF and say, well, is the location we identified for this species in the student paper within the realm or within the range of species that are found on GBIF? If it is, okay, that's like in the plus column. Let's keep looking at this. Maybe this really is a species occurrence. If it's not, maybe flag it and save it for another time to look at, more closely. And I want to be clear, like, I wouldn't ever put out a list of species occurrences generated through computational methods without looking at every single one. So what I want to do is have a tool that will help me sort through these papers and just show me that text. Show me that sentence or that paragraph that you think with 90% certainty is a species occurrence. I'll look at it and say yes or no, right? I don't want this to be making decisions for me. I just want it to help me sort through all of this text, because I can't read 445 student papers. I mean, I could before I retire, but like, I don't want this to be my only project. So this is just a little bit of an overview of what I just described, right? There are many tools and resources that we're using to try and connect and enrich the information from our student papers to help us in this determination. Like, even just trying to figure out like which of these tools should be part of our workflow for assessing species occurrences in these student papers. And I like emphasizing that, if you have a big corpus, you have a big collection of stuff, maybe it's field notes, maybe it's something else, just this first step of being able to identify taxonomic names in your text and adding that to your catalog record is hugely beneficial for researchers who want to know, should I pull this notebook or that notebook, right? Just that first step is important. This other stuff is gravy, but just that first part is really important work. But for those of you like me who really want to go to the next step and get occurrence records, it's been such a challenge. So here's an example back to our chitin paper. Recall the sort of tidy species occurrence in the methods section and compare that with the references section, right? Like, everything that you're seeing in this references section would be a false positive for computational approach to finding, like, there are taxonomic names there. Those aren't species occurrences, right? So how do you train a computer to tell the difference? And there are things you can, now it's actually pretty easy to parse out references sections. Like, you can get a computer to recognize, oh, this is a reference section, I'm going to ignore those pages just based on the way they're spaced and so on. It's surprisingly easy to train that. So you got to, like, okay, we're going to get rid of all the references sections. And this is where it gets into the part where we're still like, how are we going to do this? The first approach we tried is using a Python tool called Spacey. And this is a natural language processing toolbox that has really powerful trained models for doing natural language work. And one of the things that Spacey does really well is called entity extraction. So Spacey can read the text and say, and these aren't all right, but it can read a text and say, I think this is a place. I think this is a date. I think this is an amount of money. I think this is a person, and it can pull these things out for you. It can tag them for you. The other thing you can do is you can give Spacey a list, say, of genus and species names, taxonomic names pulled from GenFinder to include. So, okay, tag the dates, tag the places, but also tag, if you see any of these genus or species name, tag those for me too. That's step one. We got to locate all of the things we're interested in the text. And this actually turned out to be relatively easy. Like, it's pretty user friendly. There's a good online community. I got, this up and running in a Jupyter notebook pretty quickly, which is surprising, but it's like, these tools are built for people to use. So, well, I work at Stanford with a lot of really smart people who use Spacey for actual text analysis. And so I obviously didn't do this on my own in any way at all. But there's another tool you can layer on top of Spacey that actually allows you to explore the relationship between the entities. So in this example, Obama was born in Hawaii, and those two are related. Obama is the subject, Hawaii is the location, born is the thing that links them together. You could do that same thing, you could imagine, with species occurrence records, right? This thing was at this place at this time. So that would involve using Prodigy as a tool to train a model specifically to look for species occurrence records or mentions in text. And we've been trying that, and we've had some success, but I would say that my biggest struggle in trying to do this work is just making the time. Like, this is not actually my job. So I think, we see a lot of, but this is where we're like, okay, there's a lot of potential in computational tools. Keep chipping away, keep chipping away. But the reality is that language modeling is moving faster than the speed of librarianship. Like, a colleague of mine who I've been partnering with on this work, on the student papers, sent me this paper over winter break, and I haven't read it yet. But the gist of figure 1 is that, like, entity recognition and relation extraction, which is what we're trying to do with Spacey, is already now like 2 steps behind what they're working on now. So it's like, what we really need to be successful in this work is to get some dedicated staffing around, like someone who's actually trained in computational methods and data science to really get into this. So this is crazy. Well, I mean, this is just funny. I asked ChatGPT last night to describe how I might go about finding species occurrences, and it gave me a kind of a non-answer, but it mentions extracting species using named entity recognition. Like, that's pretty cool. I didn't even mention that. Anyway, we've put some internal library funding into doing short sprints with some of the software developers who normally develop the infrastructure that the library runs on, who have a side interest in doing sort of more research-based stuff. But what we really need is it, like I said, a dedicated person who can at least develop a training set where he goes through 100 papers, you tag everything that's actually a species occurrence, and then you can use a model, train a model to run on that. Like, that we really need to get that done. But I don't think it's unrealistic to be really optimistic about the pace with which large language models are coming along that you can use that are untrained. So this paper is basically using almost untrained models. You have to put in a few inputs, but I mean, it's mind-blowing. But I'm very optimistic, like I said, about the potential for computational methods to be used to extract these species occurrences from texts like student papers and dissertations. Melissa has now scanned over 150 dissertations from our collection at Hopkins, which goes back to 1892, field notes, and so on. And then one last treat I would share, because I promised Rebecca I would, is just, I've been doing a little bit of testing with handwritten text recognition. And when I first tried this out a few years ago, when I got to Hopkins, the main game was Transcribus, and you had to train it on a specific handwriting. And like, the way the field notes work on the cruises is that every day a different student does the log, and there's no training it. It just is impossible. So I put it down, and I just said, not there yet with handwritten text. And then a few months ago, I was like, because through the student paper project, we're getting, Google Cloud spun up, and we're getting accounts set up, and we're starting to use these tools. I thought, gosh, what would happen if I put some of our handwritten logs through a completely untrained vision model for handwritten text recognition? And I consider that cursive to be fairly challenging. I mean, maybe it's because we don't do cursive much anymore, but it's kind of hard to read. So I put it through a Google Vision API. There's no training. And this is what I got out. And in the short time we had, you're not going to be able to get like a super robust comparison, but this blew me away. I mean, I was really surprised at how good this is. To my eye, it's almost as good as OCR is doing with our typed text. I mean, this is just like, so this is another thing we're going to be playing with over the next year or so, is really exploring the extent to which the handwritten text recognition tools have come along. Because it's not just the Google Vision API, Amazon has a handwritten text recognition API, Microsoft also has one. So there are all these tools that you can imagine if you could use all three transcription platforms and then deduplicate them, put them together. Like, it could be a really powerful approach. I see a lot of potential for that. And so, later on in this same log, the student, Mary, happens to mention all of the species they saw along the transect line that's drawn on that map right there, right? And like, that's it. That's the data that we want, that we can get out if we can get this transcription to work. So just to summarize that crazy talk, we all know long-term time series are just critical to being able to explore ecological processes that are happening at the scale of climate change. Field stations usually have been in spots for a long time. The whole purpose of a field station is monitoring. So that's what we do. And we really, I think, have to go beyond just scanning things and really start exploring how we can use these computational tools to extract data that our researchers are going to use. And that's really what we've been working on at the Hopkins Marine Station. So I'll just finish up by acknowledging all of these amazing people who have either helped directly or indirectly by doing infrastructure at the university, our Miller Library team. And just none of this work is done by me alone. So I just want to finish by acknowledging all these folks. So happy to take questions. Thanks.
Okay, thank you so much. Questions from the room? Shannon.
I'm thinking about this amazing time machine and then running that time machine forward. So your predecessor in 200 years, how are they going to be looking at data infrastructure? How's the work you've been doing changing best practices in terms of student papers, for example? Or
That's a
colleagues that
great
you've
question. So yeah, that's a great question. I love that question. So this is something that I think about a lot. A collaborator of mine and I are working on a paper that keeps getting longer and longer and longer, where we talk about a need for library collections, but it could just as easily be your collections or some other collections to be computationally accessible, right? So the process now is we put our data into the Stanford Digital Repository, and if someone wants to use that data, they go to the repository, they click on it, they download the file, and then they have to open it and look at it. And it's like, I work at Stanford, right? Like, there should be an API for that. It should all be available directly, machine to machine, right? Like, we should be providing direct access to these materials. So, and obviously there are layers where, yes, metadata also needs to be computationally available. I think about that, right? Like, we need to be thinking about, people think about iNaturalist. People are doing AI on their phones without even realizing it, right? eBird, you can record a bird and it'll tell you what it is. Like, this is amazing. Is the library doing anything even close to that in terms of thinking about how to get patrons the information they need? We are so far behind. So that's what I think about. I think about how should our infrastructure be reflecting how researchers are actually going to not only use the data, but access the data. That's one thing I think about.
I always have a question. So my question is, what do you think it'll take to get you the resources you need, or the time, like carving out your time for something like this? Because it sounds like these are like small pilot projects, right?
Yeah, it's like pilot project after pilot project after pilot project. We've thought about writing grants so we can get staffing. I think that's one way. These so far have been internally funded, which is great. That's a luxury to have. But it's I got 5 days of developer time. And that's just enough time to get AWS and a website up. Like, it's not enough time to write a language model. So I think it comes down to staffing, right? And it's usually at Stanford libraries, we get grants to do infrastructure. We don't usually get grants to do collections. Sometimes we do, but I think I feel like maybe we should be leveraging partnerships across campus more effectively. Like there's a whole data science unit going on, and getting on their radar sometimes is a challenge because they're more interested in working with maybe the medical school or something that's a little flashier than old collections. But I think leveraging partnerships a little better could help. I think that working with students could help. There are Stanford is now starting a new data science undergraduate degree. You can get a BA or a BS in data science, depending on what you want to focus on. And they're going to need data sets. They're going to need things to work on. So if they want to work on species occurrences, that'd be rad. You know, so I think it's going to take some, well, money and or creative thinking. Yeah.
And do you see other libraries or other departmental libraries at Stanford interested in doing some of this kind of work? I assume there's like potential. Right?
Yeah, I mean, that's the fun part is I'm so spoiled by my colleagues because they're all so proactive and curious and smart, and they're all super busy like we all are. But you know, early in the work, Quinn Dombrowski and Scott Bailey, they work in the Center for Interdisciplinary Digital Research, which is like a sidearm of the library that's in collaboration with the humanities group at Stanford. And they're the ones who really got me into natural language processing. So much of this work has been collaborating with other folks in the library who actually aren't librarians, but are technologists or developers. Yeah.
Hi, exciting stuff. Thank you for
fun.
sharing. You talked about the Stanford Digital Repository.
Mhm.
What is that and how well does it work for you?
It's a great question. So Stanford Digital Repository is what I would call a generalist repository, meaning it can take digital files of any kind. So it was mostly, this is something I learned after I came to Stanford, it was mostly built for holding library collections. So we have something like 40,000 books that it's through our partnership with Google Books. We have hundreds of thousands of images scanned, thousands of maps. Like it was really meant for that with a very general metadata scheme. And now more increasingly, of course, we're starting to take in data sets from researchers on campus, especially the data management plan requirement from the National Science Foundation in 2011 really started pushing people to having to make sure their data was publicly available. So it can take any kind of data, but the metadata in its current instantiation is very limited. So for example, if I wanted to include a taxonomic name in the metadata, like you might have in Darwin Core, I can't do that. That has to be in like a notes field. So I'm actually exploring standing up my own biodiversity catalog for Stanford because our existing repository just can't handle the data in the way that I think it's best suited for. But SDR has fMRI data sets from the medical school. It has scanned stuff from our special collections and archives. It has photographs from students who upload, like we had a Black Lives Matter memorial outside in the Oval, and someone went and took a bunch of pictures, and Stanford, we accessioned them into the repository as part of our cultural history at the university. So it's really a mixed bag of all different kinds of things.
Sorry, just to follow up. It sounds like that's something that was built for and by Stanford. Is that correct?
It is. So it was built by Stanford. It is actually the underlying infrastructure is open source and was built collaboratively with teams at other universities across the US, and I think even into the EU and Canada. So it's not an infrastructure that's unique to Stanford, but we helped to build it. Yeah.
Any other questions from the room?
All right. Well, oh, there's one online. Hold on.
Oh, there's
Thanks,
people
Maricela.
online.
I won't tell you how many. Okay, so this is from Maricela. Maricela says, have you faced any objections from people that might be wary of publishing occurrences from student reports that don't necessarily have physical evidence like a photo or a collection specimen?
Oh, I've definitely heard that.
And I can ask the second part after if you want to
Yeah.
address that first.
Yeah. No, I mean, I've heard that, and I think that's perfectly fine, right? I think the important part of what I'm doing is full transparency. So if I ever do get a stack of species occurrences that I trust and I upload them into GBIF or wherever, in the documentation, it'll say exactly where these came from. It'll cite a paper or a white paper that describes how we did the work. And it will say very clearly, like, these are from student papers and you can use them or you cannot use them. And that's up to you, and I'm not offended either way. I was talking earlier with the students at lunch, and I said, like, one thing I really love about my job is just creating opportunity. Like, I'm just going to get this stuff and put it out there. And if folks use it, great. If they don't, it's okay.
Great. Marcel also asks, what other aggregators or databases besides GBIF have you considered for increasing awareness and availability of the student report collection?
Let me, so availability and discoverability are different things. I'll have all of the data in Stanford Digital Repository because it's really a preservation platform that I trust, but I'm realistic about the fact that people don't go there ever looking for data. Like, so I think it's important to put the data where people are going to find it, and that's where GBIF and OBIS come in. So I think primarily those are the two places that I would put species occurrence records. Like, I have oceanographic data too that is in Stanford Digital Repository, but if I could get to be a part of a federated catalog like Data One or something like that, where people go to discover oceanographic data, that would be ideal. So it varies by data set.
Thanks, Maricel, and thank you all for your questions.
All right. Well, thank you so much, Amanda. Let's give Amanda a round of applause. So I think there are lots of possibilities for collaboration, and talking more with Amanda about those possibilities is something we can all do at 4:45 on the L3 West balcony. Bring a couple dollar bills to get yourself some drinks. Oh, Venmo. Actually, just bring yourselves and your phone, and and we'll see you there. Thanks, everyone.
Thank you.